Okay, hi everyone. I'll try to protect my voice because I'm dealing with a bit of sore throat, but okay. Okay, so today I will be speaking about a pathogen that is maybe not as popular as Pseudomonas or the escape pathogens, but it's pretty widespread, and that's Giardia duodenalis. And what we're trying to do is to provide additional tools for public health practitioners so they can do their work a little bit better. So Giardia is really widespread and it's not only on lower middle income countries where it's usually endemic and it may cause like chronic malnutrition and even failure to thrive, but it's also pretty frequent in high income countries because it's pretty easy to get infected through any kind of contaminated water or food. So it's associated with waterborne outbreaks. And this is because the route of transmission is pretty easy. It's just oral fecal transmission. So anytime you get infected, the parasite will multiply inside your intestine. It will not invade your mucosa, so it's pretty unlikely to kill you, but it will be pretty easy to spread. Um, Giardia, despite being an eukaryote, it has some similarities to bacteria. I work mostly with bacterial genomics, so this is good for me because I can use most of the tools that I feel familiar with. And it has a bigger genome, it's 12 megabase pairs. But despite having multiple chromosomes and several nuclei, because it can have up to 8 different nuclei depending on the life cycle, it doesn't have a lot of introns. It doesn't have mitochondrial DNA. And all of these chromosomes seem to have a high identity. So they seem to have like the same sequences that makes it a little bit easier to work with. What's the issue in a public health context is that it's pretty difficult to culture. You usually require an animal model, and this takes time. So healthcare practitioners usually just do PCR to detect it, and they don't go beyond that. And even if you want to do like a public health investigation or surveillance, the only method that is available is just basically PCR and genotyping to analyze polymorphisms in 3 different genes. And this basically does not have enough resolution for something that is so widespread. So, as you probably have heard in ISMB, a lot of people, not only on the bioinformatics side, but also on molecular biologists, are working on how we can get complete genomes or at least draft assemblies from metagenomic sequencing. So we believe that in the coming years, we'll have way more whole genome sequence data for Giardia. And we decided that let's just bring something that we normally use in bacteria, that's the core genome MLST schema, where we identify all the coding sequences that are shared inside a population and try to propose something for people that are interested in this pathogen. So the first thing that we did is that we just look for all the available sequences in public repositories, and unfortunately, we couldn't find that many. We found only 125 isolates because these are amenable to analysis with bacterial genomics tools. We use Bactopia and assemble them into draft assemblies in FASTA file, but because we want to make sure that we have high quality data input to make our schema generalizable, we filter them out according to quality. Then I'm just going to point out the few major points of the pipeline that I developed. And those are we use Chewbacca, which is not only a tool that has a pretty cool name, but it also combines the functionality of identifying coding sequences for many different organisms in the life tree. It can use bacteria, archaea, fungi, and even like parasites. And it also determines the alleles inside a population using just simple blasts with a modified score ratio. And because we couldn't find a lot of genomes, ideally we would like to work with hundreds or thousands or even more, we decided to do cross-validation and just try to find a core genome inside a training set and then find what is the intersect or the common between all the iterations that we did. Further down the line, we want to do something that is a little bit more informed by the phylogeny, but that's just the final step of the project. So Chewbacca is really nice, but unfortunately it's written in Python and you have to do every step manually. So we decided to incorporate all of their functionality inside the Nextflow. So for now, and I'm to make it like generalizable and extendable to other pathogens, people just have to create a sample sheet with their input FASTA files and some parameters for their particular organism. And then the pipeline will basically do everything for you. How many iterations do you want to do of this core genome MLST caller? And just results, first thing we did is that even though we have a small sample size, we had to filter out some of them because they were pretty fragmented genomes. Some of them were dated from 2004, 2008. So probably it's a matter of the sequencer. But we found that some quality metrics like the N50 were pretty small. Some of them had a lot of contigs, or they were probably contaminated because they didn't match their reference genome length. So we just had to scrap them. But today we're trying to propose that we have a new CGM listed schema for this eukaryotic parasite that contains 4,338 loci, and just in general, one of these locus will be between 500 and 1,500 base pairs, and we'll have like 10 of 11 different alleles inside the population set that we studied. We also decided to do a little bit of annotation as quality control. Let's make sure that the coding sequences that we identified are actually part of the proteome that is described for Giardia. So first thing we did is that we used the EGNOC mapper, which is an orthology search tool that will tell us, like infer is not experimental evidence, but it will tell us what is this coding sequences, what are these coding sequences likely doing. So we identify that most of them are either participating in metabolism, cell reproduction, cell viability. And these are the things that we expect that a normal species will have in common. Like some other things like a specific niche adaptation are probably not part of the core genome. Furthermore, we blasted everything and tried to align it against a curated, manually curated reference proteome, because we know that Giardia, we don't know the function, but they have a lot of cysteine-rich proteins. So we identify a lot of these cysteine-rich proteins, as you may see in the table on the right. However, because we're not working with a model organism, we don't have a lot of annotation for some of these hypothetical proteins. So we see that a bunch of them continue to be uncharacterized. Finally, to do some additional quality control, we decided let's go back to the genomes that we excluded and see if we actually find these coding sequences inside them. And as you can see on the box plot and violin plot on the left, the lower bomb are the genomes assemblies that we ended up excluding because basically they don't present a good coverage of these loci. And unfortunately, we would like to have a little bit more genomes, but we are just currently representing the diversity that is available in repositories. So this is a minimum spanning tree that is showing each node is either a single isolate or isolates that had less than 100 alleles in difference, so they were collapsing to a single node. And we have a pretty great diversity. We see that the branches are separating these isolates from ranging from at least 1,000 differences in alleles to maybe 2,000. That's pretty good. However, most of these parasites were isolated in BC, where I'm coming from. So we would like to see if maybe we can get our hands in a little bit more diverse population set. So conclusions of this work is that we have designed a customizable Nextflow pipeline that it's based on Chewbacca and has some accessory tools, and that we hope to make these readily available. So maybe public health practitioners and epidemiologists can use these for their particular pathogen of interest. And we are proposing a novel subtyping schema for this parasite that basically, with the assemblies that we were able to include, is representing all the diversity that is currently available in repositories. So thank you very much. I also have a poster in this if you want to come and talk. Thank you.